Hello, welcome back to my YouTube channel. My name is Venon Sofa, and in today's video, we're going to talk about beta oxidation. But before we go further, please don't forget to subscribe to my YouTube channel. Make sure you like and also don't forget to comment. Tell me what you think about this video. I would really appreciate that. So let us dive into this topic. So when you talk about beta oxidation, what are we talking about? What comes in your mind? So when you talk about beta oxidation, we're simply talking about the breakdown of fat acids. So beta oxidation is a process by which we break down fat acids so that we can produce acetyl-CoA. So we are breaking down fat acids for us to produce acetyl-CoA. So the end product of beta oxidation is acetyl-CoA. In other words, we can define beta oxidation as the oxidation of fat acids on the beta carbon atom. This results in the sequential removal of a 2-carbon fragment acetyl-CoA. So like I said, the product of beta oxidation is acetyl-CoA. Now, when does beta oxidation take place? Beta oxidation, it takes place when we want acetyl-CoA in the body. So in other words, it takes place when you're starving, during prolonged starvation. Because during prolonged starvation, we don't have glucose. So the body will start using fat acids to produce acetyl-CoA. Now, where does it take place? The location of beta oxidation, it's in the mitochondria of cells. Primarily, it's the liver cells and the adipose cells, the fat cells and the liver cells. But it can occur in other tissues as well. What you shouldn't forget is that this process takes place in the mitochondria of cells. Now, when we talk about beta oxidation, it actually involves two stages. The first stage that we can look at is the activation stage. And the second stage is beta oxidation proper. But something that you should take note of here is that fat acids, they are produced in the cytoplasm. So fat acids, they are found in the cytoplasm of the cell. So we have to transport fat acids from the cytoplasm to the mitochondria because I said that beta oxidation takes place in the mitochondria. So for beta oxidation to occur, we must transport these fat acids that are in the cytoplasm into the mitochondrial matrix. So for us to transport these fat acids into the mitochondrial matrix, we must first activate them. So activation of fat acids takes place in the cytoplasm. And then when they are activated, they can be transported into the mitochondria, and that's where beta oxidation will be. Now, let us look at what I'm talking about. So what we are looking at here, this is the activation stage. And you can see that this is a fat acid chain. Now, when you look at the fat acid chain, you're going to see that we have got a carboxyl group, and we also have the carbons. So this carbon right there that you can see there, this first carbon, we call it the alpha carbon. Then we have got the second carbon, which is called the beta carbon. So beta oxidation, it's oxidation that is taking place at the beta carbon. So in beta oxidation, what do I mean? The oxidation reactions will be taking place at the beta carbon right there. So this is the alpha carbon, this is the beta carbon. This is the carbonyl group. So when you talk about beta oxidation, the reactions will be taking place right there. That's why it's called beta oxidation. And I'm pretty sure that you're familiar with or you know what oxidation is. Oxidation, it is simply loss of hydrogen ions, or we can say loss of electrons. That's oxidation. So oxidation reactions will be taking place right there. Now, what's going to happen in the activation stage? So the activation stage of beta oxidation, what's going to happen is this. And remember, activation stage is happening in the cytosol of the cell, cytoplasm. So the first thing that's going to happen during the activation stage of the fat acid is that we have the fat acid right there in the cytosol. We have an enzyme which we call acetyl-CoA synthetase. also known as thiokinase. So this enzyme is very important. Now, here what's happening is that we are getting an AMP molecule and adding it to fat acid. And then we are producing pyrophosphate. PPI means pyrophosphate. So in the first stage of activation, we are going to break down ATP and we're going to get AMP from ATP and add it to fat acid. And then we're going to release a pyrophosphate. The enzyme that is catalyzing this reaction is called acyl-CoA synthetase or thiokinase. Now, when we add AMP that we've gotten from ATP to fat acid, we are going to form a molecule called acyladenylate. So acyladenylate, this is a compound that is going to be produced. Now, when we get AMP from ATP and add it to fat acid, we're going to form acyladenylate. So you can look at this. This is acyladenylate. It's having AMP. Then the byproduct is pyrophosphate. Now, pyrophosphate can be further degraded or be broken down to two inorganic phosphates by an enzyme which we call pyrophosphatase. Now, acyladenylate, what's going to happen next? The same enzyme acyl-CoA synthetase or thiokinase is going to remove the AMP and replace it with coenzyme A. So we have acyladenylate right there having AMP. We're going to remove that AMP and we're going to replace it with coenzyme A. So you can see right there what's happening. Here we are having coenzyme A, there we add AMP. So we have removed the AMP and replaced it with coenzyme A. Now, what do we call this molecule? This molecule is called acyl-CoA. Remember, these steps are happening in the cytoplasm of the cell. Now, from there, we have formed acyl-CoA right there. This is acyl-CoA. Now, why have we formed acyl-CoA? Acyl-CoA, it is an activated form of fat acid. So we have this fat acid and we want to activate it. How do we activate it? By turning it into acyl-CoA. But why are we forming acyl-CoA? When you look at the mitochondria has got two membranes. It has got the inner mitochondrial membrane and the outer mitochondrial membrane. But what we are looking at right now, this is the inner mitochondrial membrane. Now, the inner mitochondrial membrane, it is impermeable to fat acids. So fat acids cannot pass through it. Okay? Pure fat acids cannot penetrate the inner mitochondrial membrane. Therefore, when we make acyl-CoA, we must convert it to another compound which can cross the inner mitochondrial membrane. So we have made acyl-CoA, but it cannot penetrate the inner mitochondrial membrane. So we must convert it to another compound which we call acylcarnitine. So you can see we have acylcarnitine right there. So acyl-CoA must be converted to acylcarnitine because acylcarnitine can cross the inner mitochondrial membrane. Now, how do we convert acyl-CoA to acylcarnitine? We have this enzyme right there on the cytosolic side of the inner mitochondrial membrane. So this is the inner mitochondrial membrane. Now, right here outside on the inner mitochondrial membrane, we have an enzyme that we call carnitine acyltransferase 1, also known as carnitine palmitoyl transferase 1, CPT1. That is the common name. So the common name for this enzyme is CPT1, carnitine palmitoyl transferase 1. So carnitine palmitoyl transferase 1, it is found on the cytosolic side of the inner mitochondrial membrane. Now, this enzyme, what is it doing? This enzyme is going to catalyze the removal of coenzyme A from acyl-CoA. And then it will catalyze the addition of carnitine to acyl so that we make acylcarnitine. So in simple terms, It's removing coenzyme A and replacing it with carnitine so that we make acylcarnitine. Okay, so carnitine palmitoyltransferase 1 or carnitine acyltransferase 1, it is converting acyl-CoA to acylcarnitine. How is it doing it? By removing coenzyme A and adding carnitine. Why are we making acylcarnitine? Acylcarnitine is important because it can cross the inner mitochondrial membrane into the mitochondria. Remember what I said earlier when I said beta oxidation proper takes place in the mitochondria. So acylcarnitine, it can cross the inner mitochondrial membrane via this translocase protein. So we have got a translocase protein in the inner mitochondrial membrane. It's going to transport acylcarnitine into the mitochondria. Now, when acylcarnitine enters the mitochondria, on the matrix side of the inner mitochondrial membrane, we have got another enzyme. This enzyme is called carnitine acyltransferase 2 or carnitine palmitoyltransferase 2, CPT2. Carnitine palmitoyltransferase 2 or carnitine acyltransferase 2, this enzyme, it's going to convert acylcarnitine back to acyl-CoA. Okay? The reason is simple because acylcarnitine cannot be used in beta oxidation, but acyl-CoA is what is going to be used in beta oxidation. The only reason why we converted acyl-CoA to acylcarnitine is because acyl-CoA cannot cross the inner mitochondrial membrane, but acylcarnitine can. That's why we converted to acylcarnitine, so that acylcarnitine can enter into the mitochondrial matrix, and then when it reaches here, acylcarnitine will be converted back to acyl-CoA. There is this enzyme, carnitine palmitoyltransferase 2. What is it going to do? It's going to remove the carnitine and it will add the coenzyme A. So it will form acyl-CoA. And what you should remember that the coenzyme A that we are adding here is different from the coenzyme that we removed there. So in the cytoplasm, we have coenzyme A's. Same applies to the mitochondria matrix. So this coenzyme, I don't think that the coenzyme A that we removed outside there is the same one that we have added here. No. Here we have got a pool of coenzyme A in the mitochondria matrix as well as in the cytoplasm. So CPT2, it is converting acylcarnitine to acyl-CoA and releasing carnitine as a byproduct. So carnitine will be transported back to the cytoplasm by the same translocase enzyme. So translocase enzyme is transporting two things in opposite direction. It is transporting acylcarnitine into the mitochondrial matrix and carnitine back to the cytoplasm. Now, when acyl-CoA enters, it will enter beta oxidation. So what I've explained right here, acyl-CoA being converted to acylcarnitine, then enters mitochondrial matrix via translocase enzyme, and then acylcarnitine will be converted back to acyl-CoA and release carnitine. Carnitine will be taken back by the same translocase enzyme. What I've explained so far, right there, that's what we call carnitine shuttle. So when they ask you to explain what is carnitine shuttle, please just look at what I've explained. Acyl-CoA to acylcarnitine, and then into the mitochondrial matrix by translocase enzyme, and then CPT2 convert acylcarnitine to acyl-CoA and carnitine. Then carnitine will be taken back by the same translocase enzyme, and the cycle will repeat. That is carnitine shuttle. Okay, so carnitine shuttle, this is a system that the mitochondria uses to transport fat acids into the mitochondrial matrix. It's very important. So what was happening outside the mitochondria? Okay, the conversion of a fat acid to acyl-CoA, that is activation stage. We want to activate the fat acid because I said that acyl-CoA is an activated form of fat acid. Okay, now what's going to happen in the mitochondria now? This is beta oxidation proper. Now let us look at beta oxidation proper. So we have formed acyl-CoA. And remember what I said, in beta oxidation, what's happening is that we are having oxidation reactions at the beta carbon. So let us look at these reactions now. Let's look at beta oxidation. If they ask you, where does beta oxidation occur? It's in the mitochondria. What I've explained so far is not beta oxidation. That was simply activating fat acids. Now, what we are looking at now, what we are going to look at, that is beta oxidation. So beta oxidation occurs in the mitochondria. So this is happening inside the mitochondria, in the mitochondrial matrix. So we have got the fat acid right there, and our acyl CoA has entered the mitochondria. So now let us see what's going on. So the first step of beta oxidation, this is the first step. We have an enzyme which is called acyl CoA dehydrogenase, and we have a fat acid right there, which is acyl CoA. So what's going to happen is that, so we are going to lose that hydrogen ion to FAD, so that we can form FADH2. Now, when we lose that hydrogen ion, what's going to happen? We are going to have this molecule that we call trans delta 2 eno alcohol A. It is called trans because we are going to have a trans bond. So when you say trans, what do we mean? Or trans bond? We simply mean that you have got hydrogens on opposite sides. So when you look at this, there's hydrogen on the other end and hydrogen down here. And then there's a double bond here. This is a trans double bond. If you have got hydrogen there and hydrogen down there, if hydrogen is on the same side, so you have got two hydrogens on these carbons and the hydrogens are on the same sides, that is called cis double bond. Okay, so imagine if your hydrogen was down there, this hydrogen, if it was down there and that hydrogen is down there, or imagine this hydrogen was on top and this on top, they are on the same side, that would have been called a cis bond. But since they are on opposite sides, we call it a trans double bond. Okay, that's why you're seeing trans in the name of this molecule. The other delta 2, it simply means that this bond, it is at carbon number 2. Okay, so that's what it simply means. So this molecule is called trans delta 2 eno alcohol. So the first step of beta oxidation, acyl CoA is converted to trans delta 2 eno alcohol by an enzyme called acyl CoA dehydrogenase, and we're losing hydrogen ion to FAD to form FADH2. This FADH2, we know that it can go in electron transport chain, and then we can produce 1.5 ATP. What's happening in the second step? In the second step, we're going to convert trans delta 2 eno alcohol A to a molecule called beta hydroxyacyl CoA. Now, what enzyme is here? The enzyme that is here is called eno alcohol A hydratase, or you can call it delta 2 eno alcohol A hydratase. Now, what is simply happening here is hydration. That's why this enzyme is called hydratase. What do we mean hydration? We are simply adding water. So we are simply adding water here. So you can see, you compare this molecule to that molecule. Look at the beta carbon. This is the beta carbon. There is hydrogen, there is OH. So that H is coming from H2O. So we are adding water. And look at the alpha carbon. We have got another hydrogen now. That H is also coming from water. So in step 2, we are adding water. The enzyme catalyzing this reaction is called eno alcohol A hydratase. Now, in step 3 of beta oxidation, we're going to convert beta hydroxyacyl CoA to beta ketoacyl CoA. Now, let's see what's happening here. In this step, what's going to happen is oxidation. In the first step, what happened was oxidation. Why are we saying it's oxidation? Because we were losing hydrogen ion. Oxidation is simply the loss of hydrogen ion, or you can say the loss of electrons. So remember, we were losing hydrogen ion, which was going to FAD, so that we form FADH2. Remember this H2, where is it coming from? It's coming from this acyl CoA. So in step 1, we had the oxidation. In step 2, we had hydration, adding of water. In step 3, what's going to happen is also oxidation. So we are going to lose that H. If you look at the beta carbon, we have got that OH. That's an hydroxyl group. We are going to lose that H there. Where are we taking it? We are taking it to NAD positive so that we can form NADH. So what's happening here is also oxidation because beta hydroxyacyl CoA molecule is losing a hydrogen ion. And that hydrogen ion is going to NAD positive so that we form NADH. So step 1 is oxidation, step 2 is hydration, step 3 is oxidation. The enzyme catalyzing this reaction is called beta hydroxyacyl CoA dehydrogenase. Now, why are we producing NADH? Definitely we know NADH is a very powerful electron carrier. So it can go to electron transport chain and will produce 2.5 ATP. Wonderful. Let's see in step 4. In step 4, we are going to break down beta ketoacyl CoA into two important molecules. One is called acyl CoA, the other one is called acetyl CoA. The enzyme that's here is called thiolase. So what's going to happen is that thiolase is going to break down this molecule to form acetyl CoA. So this acetyl CoA can go in TCA cycle and produce 10 ATP. Now, when you produce acetyl CoA, we are removing even that coenzyme A. We are removing that coenzyme A. It's here in acetyl CoA. And we're removing 2 carbons. So we are going to cut there. We are going to cut there, that bond. And then this is what will become our acetyl CoA. Now, the remainder part is going to be this. So what will happen is that we are going to add a coenzyme A to this remaining part, this remaining compound. We are going to add coenzyme A to that so that we form acyl CoA. Acyl CoA is having that coenzyme A. Acetyl CoA is having that coenzyme A. And here we only have one coenzyme A. So this coenzyme A right there is the one that we have in acetyl CoA. So for us to make acyl CoA, we have to get an additional coenzyme A. So this acyl CoA is going to be shortened by 2 carbons. What do I mean? Remember at the beginning of beta oxidation, we were using acyl CoA. So if you imagine that acyl CoA you're using at the beginning of beta oxidation, imagine, let's say, if it has 16 carbons. If it has 16 carbons, we call it palmitoyl CoA. Now imagine if it has 16 carbons and it enters beta oxidation. At the end, we produce a 2-carbon compound called acetyl CoA. When you produce acetyl CoA, it means you have removed 2 carbons from that 16-carbon acyl CoA you started with. Now, how many carbons are going to remain? It's 14. Now, 14 carbon, you add a coenzyme A, you make this acyl CoA. So this acyl CoA is now having 14 carbons. And that's what we mean shortened by 2 carbons. So this acyl CoA, it will go back in beta oxidation again, and it will enter beta oxidation, and it's going to produce another acyl CoA, and the same will repeat and produce another acyl CoA. So again, it's going to be shortened by 2 carbons when it goes back there. So this acyl CoA will go back in beta oxidation and the process will continue. So beta oxidation, it repeats until the entire fat acid is broken down into acetyl CoA. So guys, this marks the end of this video. I'm going to make another video very soon that will talk about energy calculation. How can you calculate ATP produced from beta oxidation? And also, what is the regulation of beta oxidation? I'll make another video very soon. Thanks for watching. Please don't forget to subscribe.